He specializes in different types of science, including microbiology, or hybrid microbiology, and molecular biology. He will know more about it. And please give a round of applause, por favor, un aplauso para el doctor.
Thank you.
Ese también es cofundador de Verne Bioanalytics. Para los que no lo conocen, es un lugar de ciencia bastante enorme. Así que vamos a disfrutar ahora este conocimiento.
Thank you. Hello, everybody. That echo is really loud there. So, my name is Nathan Johnson. I'm from Massachusetts in the United States in the Boston area. I'm the CEO and co-founder for Verne Bioanalytics. For those who are literary people, it's from Jules Verne, is where the first name comes from. It's always fun to see how people can pronounce that. And then our focus is on cannabis plant genetics. And so I'm just going to start off with a hashtag that says plant genetics is the topic you can spend months talking about. It takes years to master. There's a lot of information. I'm just at the very surface level trying to explain the power of doing that in terms of how to do your growth. So the hashtag for Verne is maximizing the plant growing potential by genetic and pathogen testing. And the principle behind that is that whenever you have pathogens in your crops, whenever you are growing the wrong crop in the wrong field, you're not going to fully maximize that nutrients or the output. And for cannabis, it's a quite complicated plant that we're still learning just to this day. So I know I had some background to talk about for me, but one of the reasons why I'm going to start off with my credentials is I've only been in the cannabis sector for about a year and a half to 2 years continuing on the side. And I wanted to show my credentials because they're not cannabis related, it's related to other areas of science. And so for those who are just talking about entrepreneurs or other scientists in this field that haven't talked about this cannabis yet, I am very new to the field and you can figure it out. It's all science, it's all the same kind of concepts, but it's in a field that has quite big impact that could affect patients tomorrow. You don't have to wait years to have impact patients. And so my background is I have education in Ph.D, master's, and bachelor's in bioinformatics and computational biology, as well as in microbiology and microbiology. Essentially what that means is I take computer science and lots of data and combine that with biology to try to understand things. That's essentially what that means at the surface level. And then where that is applied, at least in my history, is across pharmaceutical companies, cancer hospitals, food testing, plant sciences. It's all science, it's all data. They're all trying to understand some kind of correlative response to something. That can be how you grow your plant, it could be what you do to the plant, it could be how you extract it. All that is information to understand the bottom line of how you grow it. And then I've worked for some nice people like Harvard Medical School, Dana-Farber, Takeda, just to illustrate that people use that information all the time no matter the time. The big difference with cannabis is that this is just a new field. And so just some ideas or some perspective on how I treat this topic and how I come from it is I look at things like data. So I've identified a novel function for a gene related to deafness. I understand how to take approved drugs and repurpose them for Alzheimer's. Take a method to convince your immune system actually to cure cancer. Identify the computer that can be taught to look at 200,000 plus variables to try to understand what that means. I'll show an example of that later in my talk applied to cannabis. And then also identify just weaknesses in pests that affect soybeans. So in the United States, there's a pest that has a $1 billion problem. And that seems like a lot of money, but there's actually a $4 billion problem in cannabis on a viroid that we're detecting. And so this pest can just cause huge damage if you don't know if it's there until the end of the cycle of the plant. So enough about me, let's talk about science. So from talking to people at this conference, What I learned really quickly is I'm the only geneticist / bioinformatician at the conference. So I took more of a basic approach trying to explain to everybody what is genetics to begin with. Sorry, 1 second. Apparently I don't think with ears. And then what is cannabis genetics? Because if you come outside from other industries, the word genetics in cannabis actually means something different. And I'll explain a little bit about what that means. So when you start talking to other people, they're going to talk about, do you have a hand mic? Or, it's not stained, but, okay, I think we're good. Thank you. Yes, that's absolutely. Yep, thank you. So what is cannabis genetics? Cannabis growing basics, so I know you've seen some talks on just how you grow cannabis, but I want to illustrate this on how you can use testing to determine how you should grow, beyond just the analytical component, which Chris did a phenomenal job, but it starts with the DNA. It starts before it gets to that point. And then tools are available to you now, so even though this is a new industry, there are lots of tools that you may think this is almost like magic, because they're able to find things well before anything actually happens. And I don't want to add science, it's not magic. I know one of my friends said that, but that's not true. So 2 basic components of genetics is that DNA is the blueprint for all organisms, and DNA is inherited. And those are 2 very, very important concepts, because the amount of information that is in DNA is astronomical. If you were to take open one of those cells in your own body, pop it open, and spread out the DNA, it would be taller than me. That's how much information is there. On top of that same DNA, you have over 200 different types of cells in your body that do different functions. So you have lung cells, immune cells, whatever you name it. There's over 200 different functions of cells that do things. All use the exact same DNA, and it all determines what parts are used or what parts are not used in different contexts at different time points. And on top of that, DNA has a different context or different structure than what we think of in terms of our language. So the English language and the Spanish language have about 26, 27 letters. DNA only has 4. So I have 4 letters that are able to figure out different biological components that explain the rest of the biology for that individual living cell. And then using the context of humans, because we like to talk about ourselves, that if you were to think about a sentence or a word, 3.2 billion long is how long that word would be in terms of the information. On top of that, since you have both 1 copy for your mom and 1 copy for your dad, that means you have 6.4 billion nucleotides of information that you have to sift through, and that's how your body works. Same thing goes with cannabis. There's nothing different. The only big difference is that cannabis is 15 times more variable than people are. So if you were to take any 2 persons, anybody here, there'd be about a couple mutations every 1,000 base pairs. In cannabis, it's 15 times more that, which means there's 15 times more variability in how you grow and how you determine your crop. So the reason why I point that out for cannabis is that if you were to pop 1,000 seeds, you're going to see a huge variety in how you grow. And I will explain to you some of the techniques that the community uses to get around that, but it's still a consistent problem that a lot of people still have to this day. And then talking about DNA inheritance. So this is important because things you do to the plant at one generation can actually propagate downstream. There are lots of studies that look at this. This is around epigenetics as a component of science. I'm not going to talk about that because it could be a whole talk. But there are things that we think about in cannabis that get propagated on how you do these things, which are not studied, as Chris points out. There's lots of basic science that's not fully understood in cannabis yet. But in terms of DNA inheritance, if you look at these Going Bears type illustration where you can see how the colors every time you cross different plants, you can see how everything gets rearranged every single generation. You can see quickly why you would see variation from generation to generation. And there are ways to get around that. So the bottom line is, why is genetics important? And so this is an illustration on corn. So corn, about 7,000 BC, this is actually how big it was. It was not very big, wasn't very tasty at all. 2014, much different in terms of quality of nutrition, in terms of quality of what we can do with it. Primary reason of this is because of different genetic techniques. For example, molecular tagging and molecular testing that you can identify what traits you want to have from generation to generation. And so you can see how the genetic makeup can have profound importance on your final product. And this is just starting to be realized in cannabis. It has barely been tapped at all. And there's very few companies in the world that are touching this at this very moment. So now we're going to talk about cannabis genetics, particularly in terms of what that term means. So in genetics, I already talked about it from a scientist perspective, it talks about DNA and inheritance. But from a cannabis community perspective, it actually means quality of plant. It doesn't actually mean, it means kind of the same thing, but not truly the same thing. In their definition of quality of plant, you ask 10 different growers, you're going to get 10 different explanations as to what that definition means. And so if you're considering your grow, you're considering what to purchase, you're considering what seeds you should purchase from or who you should talk to, realize that you may get 10 different answers if you talk to 10 different people. So it's really critical for you to be critical of what they mean by that and ask them a lot of questions. And so on top of that, because we know cannabis is used for so many purposes, what is the maximum yield? For example, flower or CBD or THC is the most common, but fiber and all these other components interplay more or less depending on those characteristics. But the main point of these pictures on the right-hand side in terms of taxonomy and hemp of marijuana is that when you look at the DNA level, there isn't something that says, hey, I'm hemp, hey, I'm marijuana, because that definition is based on that THC marker, which in the genetic world is essentially a phenotype. It's a physical representation of that data. But you can get hemp and marijuana from all those different taxonomies. You can get it from sativa, indica, regular, depending on the components, which is a challenge because some of the community uses these terms interchangeably without understanding the physical context of what they mean. And not just these DNA markers, but also the regulators sometimes don't have that understanding, that it's truly not based on a different definition that honestly I think is not even worth it. Before talking about cannabis growing, you have to understand how cannabis grows. So all plants have a slightly different cycle. There are lots and lots of different ways on how you understand this and study this. But in terms of cannabis, these are basically the 5 steps in terms of how it's grown. And these are really, really important because depending on what you're doing with cannabis, you may actually want to extract or do different things at these different cycles. So germination simply means you're just going to have water nearby. It's going to uptake that. It's going to crack the seed open. Lots of different techniques around that. It takes about 1 or 2 weeks. Your seedlings, which a lot of the testing I'm talking about can happen at this stage, because the most important thing is that you're looking for DNA. Leaves is a really good place to find DNA. And at this stage, it'll be about 2 or 3 weeks. So if you see in the upper left corner, let me get a PowerPoint here. Because it's important to highlight these leaves here. So you see these 4 leaves right here. These 2 are cotyledons. These 2 are going to be your primary leaves. These 2 are going to actually drop off after this stage. And so if you want to do testing, you might as well test on something that the plant's going to throw away anyway. And then after the seedling stage, you're going to have what's called a vegetative stage. This is going to be one of the controversial areas of cannabis growing, because how long you keep it in vegetative, how big you want it, what you do with it in that cycle can drastically change the cannabinoid output. And you can actually have plants that are stayed in vegetative state for years. There's one grower who's done it for supposedly 20 years, just constantly taking plant leaves off and just making more and more clones. And then the flowering stage can be 6 to 8 weeks. And that is what most people worry about, is they want to get those nice big buds, those big flowers. And then you harvest it at that point. So as I mentioned, in terms of how you grow cannabis, what you want to make it, you basically need to have 3 different considerations when you're thinking about this. If you want seeds, i.e. you're going to be a producer, you're going to want to keep pop-out pushing going, you're going to need both male and female. There's a 3rd component. There actually is a hermaphrodite state in cannabis, which means that you have pollen that actually will actually give you seeds, but it doesn't give you the actual maleness that a lot of people are looking for in terms of diversity. And so if you want flower, you do not want male, you do not want hermaphrodites, because once you start making those seeds, that cannabinoid level is not going to be produced as much. And so different techniques that screens for only female is really, really valuable on point. And then if you want fiber, some growers actually don't care if they have seeds or not in terms of their fiber, because they'll use that seeds to keep propagating them. But one thing about cannabis is if your neighbor is growing for a flower, your pollen in your field is going to affect them. And so there are some states where people are actually going to court over that problem because you can lose 40–50% of your yield just from that fact alone. And then different rules, at least in the United States, apply for different purposes on these different things. And so you have to be really considerate of those considerations as you are deciding your whole grow operation. And then your harvest time is also going to be super critical. So some of these cannabinoids that were mentioned beforehand show up at different stages of that life cycle of flowering. Sometimes they start in the really early, sometimes they end up in the later age. And depending on that combination you want, you may want to decide that harvest. So essentially what I would suggest is that for anybody who's going to grow, just don't just go into it. Do several controlled experiments to understand your plot of land as well as your environment before just jumping into this. There is many farmers, especially in Massachusetts, bankrupt because they didn't do this. They devoted their entire life savings, their entire growth to doing cannabis, and now they're bankrupt. For those people, the reason why they're bankrupt is because they got seeds that they thought were only females, and they never thought that there'd be a male in them. And it turns out about 40% males showed up and completely ruined their crop. That's actually how I joined the industry, because a friend of mine introduced me to the field because of that. So this is one of our growers. So we do a lot of testing for growers to see how well they're doing with their crop. Most of them are going to be home growers like this, just because we are in the field of R&D type testing. We're not in regulatory testing that was talked about beforehand. And that area is a little bit more gray in terms of what that means. But essentially what's going on here is if you look at these different colors, if you look at these different terpenes, and again, all those white hairs, I know some people are familiar with them, sometimes I'm not, but I still find them super fascinating. Even the color there will kind of tell you what terpene or cannabinoids are present. But in terms of the DNA composition of this, you can predict all these things, as Chris mentioned, in the vegetative state or even the seedling state, because that DNA holds the blueprints to what's everything's possible. So going back to the cannabinoid production, there are two enzymes that are required for CBD and THC. One of our simple tests we have at Verne is those enzymes even there? Are they functional? Because if they're not there and they're not functional, you're not going to have CBD or THC. This is one of my favorite slides that one of the growers in California put together. And it's just a really nice summary of all the different growing strategies that exist. This goes by the types, as well as the time of planting, harvest, consistency, cleanliness, pathogens, costs, specialists, and genotype variation. And the reason why this is important when you're considering your grow, because all these decisions make a difference at the end of the day. In terms of time, you're talking about months saving in terms of that difference. You're not talking like days or weeks, you're talking like months in terms of the difference. And so my preference, I like seed just because I'm a geneticist, I like to understand those things, but from the business perspective, what I've noticed from most of my growers is that taking clones from tissue culture derived mothers is the best approach. And I can show you a little bit about what mothers look like and what tissue culture means, because it's not really easy. It's going to take a little bit more infrastructure. I don't know if that's in plans here in Costa Rica, but that is something that works really well in the United States. But essentially what this is trying to take care of, if you remember the 15 times DNA variation, the reason why they're doing these things is to control for that. They're trying to make sure they grow consistency so they can predict what they're going to grow. And then the other thing is also pathogens. So as in analytical testing, humans are usually the main factor in terms of this problem. For example, I have a buddy, I want to use their seeds, I want to use their plant, thinking, oh, they may have viruses in their grow that may contaminate mine. And so there are some pathogens that don't show up until 3 months down the road, and therefore you lost your crop. And so this kind of testing that I'll be talking about in a little bit here is to help with those growers know what they can't see so they can take care of it before it happens. So this is an example of cloning through mother plants. It's not a very difficult process. It's a very simple process, but it's also easy to screw up if you don't do it correctly. And what I mean by that is if you take these mothers, they usually have them in an indoor grow, completely security locked, because these mothers represent their entire brand in terms of what they're doing. When you're taking your clones here, you notice they can this sharp edge at the very edge of that clone. You want that piece, and that's where all those cells are going to be nice and reproduce the roots and everything. And then this kind of cup, again, this is a home grow. Right here, this is one of our growers. Just in terms of their testing, trying to understand what the variation for their genetic line that they're working on. And so it's a very simple process. You have a bunch of mothers, you're going to clone them. But one thing I haven't talked about is how do you determine which are your mothers or which are the things that you should grow with. So in the cannabis sector, they call that what's called a phenohunt. So a phenohunt means that you take thousands and thousands of seeds and you just screen all of them, trying to understand what they make, and that becomes your mothers. That's very time consuming, very expensive, but molecular type testing can tell you what's there before it's even showing up. This is an example of tissue culture from one of our collaborators that we work with. If you notice, it's a lab setting. So these hoods here on the bottom left, you literally look under the microscope with a pair of scissors and you cut off the very tip of those leaves to get the meristome, which is the cells that will completely divide to make a full rooted plant. In the middle here, these are their tissue culture. Literally what this media is made out of is agar, seaweed basically, that gives it that really nice thick texture. And then you have different hormones and nutrients to help that plant grow. It's a very artificial setting, but if you notice here on the bottom right, look at the capacity that you can grow in a very small location. So in this one incubator, you can have 40, 50 plants being grown at once rather than a full big plant. And so this is where the tissue culture from mothers really helps, because you can massively expand your product line that's linked to that one plant. So now let's talk a little bit more of the testing and a little bit more of the science. So the genetic testing that I'm going to give 3 examples is the cannabis gender, disease resistance in cannabis, and then identifying a pathogen in a plant. So about the cannabis gender, around the seedling stages, when you can run both of these tests at the top, you can run the pathogen test at this point, but depending on the pathogen, you might not find it. But the idea is that if you see something sick, you can just run the test and know within 30 minutes if you have something present or not. So this is the magic part, because this test can be run in your car if you want to. You can run this on a hot plate. You don't need a lab to run this test. And I'll talk more about the why in the next few slides, but I just want to illustrate what this does and what's the power here. And if you look at this color change here, what's happening here on the right-hand side is that one of our growers took about 30 other plants and they screened them for male or female at day 3. And then based on this color change will change if that maleness is present or not. I'll go into the details of that later, but I just want to illustrate why this is powerful, because within 30 minutes, you know that what will take 2 months to find out within that day. Another example, so in the United States, I don't know if you have it here in Costa Rica, but I'm sure you do. Powdery mildew is a really big problem here. Once it gets to 80% humidity, powdery mildew will go everywhere. It can really devastate your crop. So you see on the left-hand side here, a little powdery, little filth here. That is going to reduce the overall yield and sometimes make you fail your regulatory testing depending on what you do. One thing I will point out here, look at the date of this paper that identified where that trait came from. It came out this last year. That's one that is really common in terms of cannabis, is there's lots of people trying to understand this plant, but we're all in a big horse race trying to understand this plant. And so as pointed out beforehand, update these different figures all the time. I have to do the exact same in terms of different genetic traits that may be interesting, because they're always being discovered and it depends on the context. But the right-hand side, this has the gene that doesn't allow that powdery mildew to actually sporulate. On the left-hand side, it does not. And so if you're trying to find a disease-resistant plant, you can know at day 3 if it has that gene instead of having to wait. And so another challenge here in the United States is what's called Hopslate and thyroid. It's thought to be the $4 billion problem, because you can have up to 40% loss of your yield. And on top of that, it has this nasty thing that it can go dormant. So you may have 10, 100 copies of the thyroid. It's not doing anything to the plant. You stress out the plant, and you got trillions of the thyroid area killing everything. And so what this on the right-hand side is showing is just showing the sensitivity in the range in terms of that testing. And so that yellowness, again, is indicating that it's found the viroid and it found it at that concentration. But like all testing, there's always limits on what it can find and what it can't find. If you notice at about 1,000 of that viroid, it cannot find it anymore. And so going back to the question about can you ever call something THC-free, it all depends on how you test for it and the technique you use. Any scientist, anybody who knows what they're talking about will always say the caveats of their testing because we all know that they exist. You could always have a scenario to make the test fail or succeed, depending on the manipulation. So let's now dive into how is this possible. So I'm going to have one slide focusing on the non-scientific way of talking about this, and the next slide talking more about what is the molecular component that's being exploited here. And so essentially we're just trying to do three different things. We're trying to find that address on that DNA or RNA that represents that phenotype. Now, if you remember your school, if you remember Punnett squares, if you remember like the little four square, you're trying to look at different combinations, rarely happens in terms of real life. Blue eyes, for example, has 100 different locations across DNA that allows that to happen. And think about that 6.4 billion nucleotide pieces, lots and lots of pieces that go together to make that phenotype happen. And so one of the reasons for having a computer science degree is have the ability to sift through all data to figure out what's the components that we care about. And so I just call it address, but it may be addresses of addresses that actually exist. And then you develop an assay to find that address and amplify it. Because that location, that piece of sequence, isn't going to be enough for us to actually visualize it and detect it. So you have to have some way to make more of it so you can find it. And then you have to incorporate a way to visualize that output in terms of that, what it looks like. So it could be fluorescence, it could be color, it could be different types of colors. So you need to have some way to visual cue to find it. So here's more of the science-y version of that same thing I'm talking about. So in this case, we're using an illustration of a gene of interest. We have a gene illustrated as these two lines here. You have these mutations and these gene variants within this gene. Identify and quantify that gene expression. That could be done through many, many different techniques. Between sequencing, PCR, all these different ways to try to figure out what that phenotype-genotype correlation is. And then you go through a series of experiments to see if you are consistent in that observation. One thing I always tell our clients is that we don't want A answer, we want B answer on what this is supposed to associate with. And so now let's look into the data of how we actually developed one of our tests here. So for those who are more familiar with science, this is what's called a transcriptome. What this literally does is it analyzes all RNA across a given sample. And so each of these dots represents a single gene or a single transcript that represents what it's doing in association with these males and females. So there's 70,000 dots here. In reality, there's going to be more than that. That's going to be limited based on what we know. And this is the RNA-seq transcriptome analysis is what this plot will look like in these next few. But the illustration, what I'm trying to show here is how you can help identify the phenotype to genotype relationship. Because on the y-axis, what's being done here across all our samples is trying to ask the question, is this correlative to that phenotype? On the x-axis, it's looking at the expression or the amount that's there. And then these color dots just kind of give you a nice threshold on what we want to focus on. And so you do a series of analyses to figure out, okay, what should I focus on for our assay? That's going to actually give us the answer that we're looking for. And so if you notice here, this is the explanation to what the circle blocks represent, is the higher male expression is the top. And so we have our candidates of male and female expressed transcripts. And so for this experiment, we wanted something that related to maleness or femaleness that is never ever expressed in any other gender, because we don't want any false positive in our analysis. So another way to look at this is also look at the variation across our data set. So one of the methods that we have at Verne is that we actually automatically pull in all publicly available data for cannabis and analyze it automatically, because I want to know what is the inherent variability across the world. Because again, cannabis is very, very variable, and I want to know consistently if we have the right answer. And so if you notice here, again, we see similar transcripts are not labeled for proprietary reasons, obviously, but the purpose here is to indicate these are our candidates in terms of that consistency on that beta value or on that variance level. So This is a little bit more complicated to explain, but for people who understand principal component analysis, what it's trying to do is it's trying to take all these values and compress them into a dot that's 2D. It's essentially taking 70,000 dimensional space and put it to 2. If you think about that, that's pretty hard for most people to comprehend. I still have trouble. But the idea is that if you compress that consistently, or there's different ways to do that, you can identify how consistent or how overlapping my samples are. And if you notice, we labeled them female and male, you can see that there actually are some males that overlap quite strongly with the femaleness of all those transcripts. And the other thing that I always get excited about is whenever we see really big outliers, like this one on the far right-hand side. Now that could be completely related to technical reasons, but more often than not, it's related to some kind of biology that's not understood. Now, since we only have one, we don't necessarily know what that means, but it is interesting and something that we can compile over time to understand what does this dot actually represent. And maybe it's some novel finding that we haven't discovered yet. But essentially what this allows us to do is help us to look at all our data and ask the question, are those RNA transcripts consistent with what we're looking at, and should we have any warning flags on that analysis? So this is an example of one of the transcripts that we identified. If you notice on the naming scheme, we have replicates across different stages of growth across the flower for the female. And then we have the male flower stage to try to determine if that's associated with maleness or femaleness. And if you notice, it's quite strongly associated with stage 1 and stage 3. There's kind of a second level here, where it goes up in stage 1, down a little bit in stage 2, up in 3, and down in 4 again. But the whole point for what we're doing in this thing is not to understand the biology, it's trying to find something that tells us it's male or female without any guesswork. And in this case, we have something that does that. Let's talk about how we actually do this. So polymerase chain reaction was talked about beforehand. Trying to get a time check. But then polymerase chain reaction, what it's trying to do is it's trying to amplify that DNA. So the enzyme that's actually responsible for PCR was identified in Yellowstone in the United States. It was actually identified in a thermal vent. Its natural living environment is 100 degrees Celsius in that area. And so the reason why the scientists isolated that is they wanted something that could actually unravel DNA, that the enzyme could still be functional. And so if you notice on the y-axis here at 98 degrees Celsius is when those DNA strands actually separate to allow that enzyme to come in and do its thing. But one thing that people won't necessarily tell you is that the machine on the left-hand side here is $20,000 or so. Most growers, most people don't want to spend that. I don't want to spend that. And so at Verne, we were trying to rethink the problem and say, hey, can we do something to get rid of that? Because I don't want to spend that money. The next question, the next problem here is, can anybody run a PCR? So one thing with PCR is that it took us 6 months to train our technicians to run it in our lab. And that's with my CSO who is over their shoulder all the time. And the reason why you have to be so careful is that you can find single molecule detection in your lab. And so if you're not careful on how you move all that plant material around, you can just contaminate it just because you put your hand over the tube, and now therefore you have a molecule. And so what this does here is you have your plant material, you have all your different components. All these things are different nucleotides to expand that DNA, and you're going to go through that denature annealing extension stage, which is what those cycles are for. Now, to talk about more of the workflow beyond just the PCR, also talking about the sample extraction process itself. So taking that leaf, popping up with the cells, having the DNA there, having it able to run the reaction, have it amplify, all those different steps require different cleaning steps because plants are notorious to have molecules that can actually ruin that reaction. And so you have to have different cleaning steps to inactivate those things. And so this is what the whole workflow looks like. It's somewhat simplified, but on the top hand side is DNA, because the RNA is a very unstable molecule. We have to reverse transcribe it back to cDNA, usually using an HIV virus. And that gives us something that we can constantly detect that's positive or negative for whatever phenotype we want to care about. And so talking about beforehand, we wanted something that was simplified, could be run by people who have no training and at low cost. I don't want to spend 25 bucks every time I run a test. I'd rather spend a couple dollars. And that's what we're going for in terms of at Verne, is in terms of that perspective. And so we wanted to have expensive equipment, get rid of, And we wanted to reduce the complexity down to a really simple protocol. And so we went with a technology called what's called isothermal amplification. Essentially what that word means is that at a constant temperature, you get amplification. And the reason why we're able to do this is because we can use different enzymes. And if you can think about it like a catalog of a textbook or a cookbook, there are thousands and thousands of enzymes, thousands of different tools that can be mixed and matched together to make different things possible at the molecular level. Bacteria are wonderful from that perspective. And so we chose a bacterial enzyme that was able to amplify at the same constant temperature, so we didn't have to have that machine. And so we could eliminate that whole machine process, but we still had a detection process problem as well as a sample extraction problem. And so we went with a method what's called LAMP. We no longer use LAMP because we did it because we wanted to make it better as we usually want to do. But for illustration purposes, we wanted to show at the molecular level what is actually happening under the hood. So you have these two different methods called LAMP and RPA. They're both isothermal amplification. They're in the market. They're used for different purposes depending on what you want to do. But the idea is that you want to make this loop structure to be able to amplify that particular region of interest. And so by manipulating this loop structure, you can get different pieces of DNA to make stable molecules that you can detect. And this is how you put in fluorescence and things like this for you to be able to detect it. RPA is slightly different. It's more designed to be like a PCR-like process. The nice thing about it, it's easy to design, but it's active at room temperature. And the reason why that's a problem, a lot of people will say, I want to put it in my hand, I want to run it, but how am I supposed to ship it to you? How am I supposed to make sure that when you're setting up your assay, you don't have it thawed and it's not active? And so after the hour of having it on the bars up there, it may be inactive. And so we wanted to have an enzyme that required a temperature change so that to be activated. And then we went to visualization chemistry. So how do you visualize this methodology that you can't see? So there's lots of different technologies out there. We decided to go with what's called phenol red, which is a pH indicator. DNA is negatively stranded, and so it will cause an acidic pH change when you get more and more of it. But there are other technologies that are out there that we considered, but for various reasons we ignored them. But the main idea is we wanted something that was crystal clear, pink or yellow, yes or no. And so this is what we did in terms of the process. This is literally our entire protocol on how you run it. You have that piece tipped, you literally punch a hole in that leaf, you swirl around in that piece of liquid for 10 seconds, you cap it, you incubate it, you're done. And the reason why it's able to do this is because we have different molecules in there to remove or inactivate things that would inactivate the solution, as well as we have molecules in there that make sure that our enzyme doesn't do anything until we incubate it. And the nice thing about the incubation temperature is at 63 degrees C, which actually a lot of ovens in your house can actually go that well. But because we wanted to standardize and make this consistent, this machine here is $50 on Amazon in the United States. So you're taking going from $20,000 to $50. You're going from $12 or so to a reaction to $4. And so therefore, it just makes it so much easier to get the technology or the information that you're wanting. And so not to just show the power of this type of technology, this type of technology is also quite sensitive on what you're looking for. And so in our case of the viroids, just to give you some perspective of viroids, if you have the numbers around here, that means you have a highly infection going on. If you have numbers around here, the numbers are starting to go up in terms of infection. If you have numbers down here, that means it's latent or it's not going to be expanding at that moment. And so for our versions 1 and 2, just make it more and more sensitive because we want to get down to the answer of can we know if it's there or not and what can we do about that. So unfortunately at the moment, when it comes to viruses and plants, there's not really any ability besides quarantining or burning. And so early detection is really the only way at the moment so that we can get rid of that. We have research under the way to try to see if we can figure that out, but it's taking time. So INTA and Rocco, we were able to convince them to run our test on their fields to see how well this testing worked across, just to predict it. And so this is essentially what we did. So these plants in the background, this is when we took the leaves. We predicted 50 cannabis plants if they were male or female, waited for 6, and then verified that it was 100% accurate. Now, I know that as we expand to thousands and thousands more plants, there's going to be certain hermaphrodites that pop in. It's going to cause some difference. But generally speaking, what you're looking for is some consistent DNA to represent that male. And the reason why we were able to do that is because we wanted a piece of DNA that was linked to making sure that plant is male. We don't want some sequence that just is inherited. We want something that actually related to the maleness trait. And so on top of that, we had students there. And so as a proof of concept in terms of explain the technology, I wanted them to run the test themselves. And so them taking this across all these plants, this is a very easy process. And then if you look on the left-hand side, this is me watching them within 5 minutes of demoing the process. They are running the test themselves. And the idea here is just to show you that there are tools available to help you understand the genetic component of your plant. There are many, many more things that we need to discover, many more things that we need to understand, but there are tools available both from chemistry and genetics that can help you understand this plant. It's not just a black box. Just because it's not a lot of science hasn't been done doesn't mean there isn't science already. And there's lots of companies like ours that is trying to invent more and more to make this more and more practical. And so just the take-home message is that use the genetic tools to grow better, grow smarter. Don't waste your time waiting for months for something to grow when you already have a tool that can give you the answer. Try to solve a problem that needs to be solved. Don't try to reinvent the wheel. And then what tools are available to expand every day across the community, the world, honestly. And then critical to be purposeful in planning your operations in terms of how you grow, indoor, outdoor, cloning, tissue culture, what you want to grow for, how you want to grow. All those SOPs need to be figured out on both your side as well as the testing side as you plan out your operation. If you do not plan, you're not going to work because this plant is really, really complicated to get what you want. It's easy to grow, but it's not easy to ramp up your production, is what I'm trying to say. Some people call it's like a weed, and it kind of grows like a weed. It's pretty resistant, but at the same time, there's lots of things that like to eat it. And so you have to be careful of how to mitigate that. And then reach out for assistance for myself or anybody else if you have questions. I always love talking to people who actually work with the plant because I always learn something new. Thank you for listening to me. I hope you enjoyed something and learned something. And I'm open to questions.
Hello, thank you for the space. I would like to ask, what would be your advice for choosing the right seed to actually get the genetics that you want? We have a vendor's database, we need the third-party analytics, or do we have some regulator certification for genetics, for example? How do we choose the right seed, which is making sure that we're getting this genetics? Thank you.
So this is a very, very good question. I don't know what the rules are in Costa Rica yet. So